And our next speaker is Deborah Chasman from University of Wisconsin-Madison.
Oh, spoilers. Here we go. All right, can you all hear me? All right, I'd like to start by thanking the organizers of the RegSys track for giving me the opportunity to present our work, and also to the ISMB Travel Fellowship for helping me to attend this conference. Gene regulatory networks connect regulators, such as transcription factors and signaling proteins, to the target genes that they modulate. Changes in regulatory networks can significantly alter the type or function of a cell, which can affect both normal and disease processes. One of the important problems in regulatory genomics is to understand what these cell type specific regulatory networks are and characterize the dynamics by which they transition between cell types. Building networks entails identifying the important regulators, which I'm showing here on this slide as X, and defining the function by which they regulate their targets, which I'm showing here as Y. So I don't think I need to remind all of you that this is a really hard problem, in part because there are many levels at which gene expression is controlled, including cis factors like histone modifications and sequence elements, and transcription factors, which are transacting factors. So the problem that we're trying to approach here is to infer cell type specific regulatory networks for multiple cell types by integrating all of these different types of regulators. Existing approaches to infer cell type specific networks can be grouped into different categories based on the types of data that they integrate and the features of the networks that they infer. There are very few so far that combine epigenetic and expression data, or that can handle multiple cell types at the same time. Multitask regression methods that infer expression-based regulatory networks simultaneously for multiple cell types have been successful in learning regulatory networks for multiple cell types at the same time, overcoming the small sample size problem by sharing information. But in our case, we find that there are many cell types for which we have only a single sample of expression data. And so our question is, how do we learn a parameterized network with so little data? We propose a new approach to infer regulatory networks and related cell types, which we call dynamic regulatory module networks. The inputs are cell type specific regulatory signals and sequence features. The outputs are inferred regulatory networks, one for each cell type. So in order to tell you about DRMNs, which is our approach, I'll start by telling you what a regulatory module network is. In RMNs, we use a module representation for gene expression. A module is defined by a set of genes that have a particular expression state, which we can interpret as a discrete level. So in this example here, we have two modules, one containing high expression genes and one containing low expression genes. Using a module representation is important because we have only one expression sample. The next component is the regulatory program. This includes the graph structure, who are the regulators, and the parameters for how those regulators predict gene expression. We represent each regulator with a vector of measured values for the strength of its signal for all genes. This is how we can incorporate chromatin level information as well as other kinds of regulatory signals flexibly in the approach. We use a simple linear regression model to predict gene expression from these feature data. And we learn one regression model per module. One regression model per module using a greedy algorithm. So dynamic regulatory module networks are a multi-cell type version of RMNs that inform regulatory programs for multiple related cell types organized on a lineage tree. To do this, we introduce another set of parameters, the transition matrices, which describe the dynamics of how genes move between expression modules between different cell types arranged on this lineage tree. We use these parameters as priors in learning the regulatory networks and the module assignments. And we do the learning of all of these networks simultaneously with an expectation maximization algorithm. I'm now going to talk about an application of DRMN that we're working on with our collaborator, Rupa Sridharan. Her lab is interested in reprogramming differentiated cells into pluripotent cells. This can be done by inducing just a few transcription factors. So we know that these transcription factors are part of larger regulatory networks, and our question is, what are these networks in the different cell types and how are they changing? We applied DRMN to 2 data sets that profiled mRNA and chromatin marks in cell types during different stages of reprogramming. One of the data sets was profiled by array and the other profiled by sequencing. Both included a differentiated cell type, mouse embryonic fibroblasts, a pluripotent cell type, either induced pluripotent stem cells or embryonic stem cells, and 1 or 2 intermediate cell types at different stages of reprogramming. So we're really interested in learning which types of regulatory signals are the best predictors of expression, and also how to represent them. I'll briefly describe how we're currently representing the signals. We have 8 or 9 different chromatin marks measured from each of the 2 data sets. For each gene, we represent the chromatin mark signal as the average signal in a small window around the gene's transcription start site. We also have a set of motif features which are not cell type specific and are based simply on the strength of motif matches near the gene's TSS. The sequencing data set also provided ataxic data measuring chromatin accessibility. We use these data to create 2 additional sets of features. One is a simple ataxic feature, which is the average accessibility in that window around the TSS. And the other is a set of cell type specific quantified motifs, where we've taken our motif features from before and tried to quantify them by using the ataxic signal under motif instances in the gene TSS regions. We evaluate DRMNs by their ability to predict held-aside gene expression. So in this scatter plot, each of the dots is a gene, and the color is its predicted module assignment. So in this result, we have 5 different modules in the DRMN. The x-axis is the actual gene expression, and the y-axis is the predicted gene expression. So to quantify this result, we first find the correlation of predicted and actual expression within each of the modules. And to get a single value for the entire result, we take the average of those correlation values. The reason that we don't use the overall correlation for all genes at a time is that those values tend to be misleadingly high. And we're interested in making sure that we're capturing the variance as well as the mean. I'll start by showing the result of using motifs to predict expression. So each panel here is the result for one of the 2 data sets, the array data set or the sequencing data set. The x-axis is the number of modules that were learned in the DRMN model. And the y-axis is the average correlation per module, so higher is better. Each of the dots here, or the different shapes, is the result for 1 cell type. So we can see that there is signal to predict expression from the motif features. It's not very high, but it's there. Now in blue, I've added the performance of the chromatin mark features as candidate regulators in DRMN. So these features definitely outperform the motifs, but we see that they do decrease in performance as we increase the number of modules. Now in red, I've added the result of concatenating the motif features with the chromatin mark features as candidate regulators for DRMN. And we see here that the combination outperforms either feature set separately. So this was our best performing feature set. For the sequencing data, we also had that access to the accessibility data measured by ataxic. Here, I'm showing the motif features again in yellow, and I've added the performance of the ataxic feature in green. So this was not a very good predictor, and this might not be very surprising just because we have only one feature to predict expression. Here I've added these purple dots, which are the result of concatenating that ataxic feature to the motif features. So it does provide some improvement. We had also tried to generate cell type specific motif networks by quantifying the motifs using cell type specific ataxic data. And we didn't, these are the light blue dots that are pretty near the bottom of the plot. We didn't see that they performed very well, and we think that it's because it's too sparse. And finally, we tried adding that ataxic feature to our existing best performer, the combination of chromatin marks and motifs. And that result is shown in light gray next to the red result, which was our current best performer, and it didn't improve anything. So it seems that the ataxic signal in this data set was redundant with the chromatin marks. So it's important to ask also, our next question was whether using the dynamic regulatory module networks was an improvement over just learning these networks separately per cell type. So we compared DRMN to regulatory module networks learned separately for each of these 3 cell types in the 2 different data sets. And I'm showing results here for the same features, the chromatin marks and the motifs. So the DRMN results are in red, and the RMN results are in blue. And especially in the array data set, the joint learning was helpful. And in the sequencing data set, we see that it was helpful especially for some cell types more than others. So the red dots coming down the top of the sequencing set are the embryonic stem cell measurements. So now we'll go over some ways that we're using DRMNs to try to learn about the underlying biology. This figure shows all of the module assignments in a learned DRMN. So each row in this figure represents one cell type, and each of the bars is one of our different modules. There were 7 modules here. Each bar stacks up all of the genes assigned to the module, where the genes are colored with their expression value. So you can see that this is a pretty decent discretization of the expression values, and also that the interpretation of the states is matched pretty well between the cell types. One way to start digging into these results is to look at the genes that are within one module in one cell type and ask whether there are enriched sequence elements here compared to the other modules in the other cell types. So just as an example, I'll show the results for the most repressed module. So this graph is showing the rows are 6 different transcription factor motifs, and the columns are the different cell types. And each cell is colored according to the strength of the enrichment of the motif element in the module for that cell type. We do see some cell type specific enrichment. We see BCL6 for the pre-IPS cells, and very strikingly we see TCF3 in the MEF and the MEF after 48 hours of induction. This seems to be consistent with the literature that we know about TCF3, which is an important regulator of pluripotency, one mechanism being by repressing pluripotency genes. I'll also show you an example from the most highly expressed modules. So here's another similar plot. We see some cell type specific enrichment patterns for some transcription factors, including MYC. MYC is a very important regulator of pluripotency, and it was one of the 4 transcription factors that were induced to do the reprogramming in this data set. But instead of just looking at each module of genes separately, we're really interested in looking at how things are changing between the cell types. So one way that we can look at changes over the reprogramming process is by looking at gene trajectories. That is, the sequence of modules that a gene progresses through the cell lineage tree. So this is an example trajectory of a gene that is very repressed in the differentiated cell type MEF at the bottom and highly induced in the pluripotent cell type embryonic stem cells at the top. We clustered genes by these trajectories to identify groups of genes that transition their expression states together. One trajectory that we found included 49 genes that are very specifically expressed in embryonic stem cells. The heat map on the left shows the module assignments, where the pink colors are the high expressed modules and the green colors are lowly expressed modules. And the heat map in the middle shows the expression levels. So these genes are also strongly enriched for TCF3 at their promoters. So to summarize, I told you about DRMN, a predictive model for gene expression in multiple cell types. We're able to flexibly incorporate chromatin information and use multitask learning and a module representation of genes to overcome the problem of having only one expression sample per cell type. We found that together using chromatin marks and motif features were the best predictors of expression, and the joint learning could outperform independently learned models, especially in the array data set, and for some cells in particular. So I'll conclude by thanking my funding sources, including the Travel Fellowship, and I'll open up for questions. Thank you.
The way you set up your model, and something you showed is that adding in the histone modifications leads to a huge improvement, and I think there's a lot of cell type lineages where it would be interesting to apply this, but doing all the histone modification experiments is going to be expensive. I was wondering if you've tried to see, is there a subset of combination of marks that can get almost all of the improvement, or do you really need everything you put in?
That's a very good question. We haven't looked at that yet, but I think that would be great to look at. Thanks.
I was wondering, in terms of the chromatin data, sort of how far out you went in the promoter region, and if you did anything outside the promoter region?
Yeah, so we've only tried like one configuration at this point. So for the array data set, it was averaging over 8 kilobases around the TSS. And for the sequencing data set, we looked at 2.5 kilobases in each direction.
Since you're doing reprogramming here, can you identify regulatory networks that show some memory from the original cells that may still be there in the transdifferentiated cells?
That's a good question. So I guess that would be pieces of the network that are constitutive across the cell types.
That never change, although they should, because they would be in the initial network, maybe they would be together, but then they separate, but they don't go together into the reprogrammed state as they should.
I see. I guess we could look at the trajectories that way, so see which in the beginning cell type, try to make a branching tree instead of a linear trajectory. Thanks.